In our scenario-based discussion, we talk about how the driver was confused about how to get the people to the other section of the bus. Two things can happen. Either the people will move by themselves, or the driver might have to force them to move from one place to another. The same is the case with diffusion. It can occur on its own and can be known as simple diffusion or non-facilitated diffusion. And it can also be occurring with the help of an outer entity's help, which will be known as facilitated diffusion. So, you must be curious about both of them. Let us talk first about non-facilitated diffusion. In this type of diffusion, particles don't need the help of a foreign agent to move from one place to another. Now, the particles are of two types: lipid-soluble and water-soluble. Before diving into these particles, let us understand the structure of the cell membrane. This membrane consists almost entirely of a lipid bilayer with large numbers of protein molecules in the lipid, many of which penetrate all the way through the membrane. The lipid bilayer is not miscible with the extracellular fluid or the intracellular fluid. Therefore, it constitutes a barrier against the movement of water molecules and water-soluble substances between the extracellular and intracellular fluid compartments. However, lipid-soluble substances can diffuse directly through the lipid substance. Now remember that we talked about lipid particles? They will diffuse rapidly through the lipid bilayer. For example, the lipid solubilities of oxygen, nitrogen, carbon dioxide, and alcohol are high, and all these substances can dissolve directly in the lipid bilayer and diffuse through the cell membrane. It is like mixing Coca-Cola with water. Try it if you don't believe me. The rate of diffusion of lipid-soluble substances through the membrane is directly proportional to their lipid solubility. Take the example of oxygen. Especially large amounts of oxygen can be transported in this way, therefore, oxygen can be delivered to the interior of the cell almost as though the cell membrane did not exist. Does anyone want to ask me what happens to water-soluble particles? Well, water-soluble molecules cannot pass directly through the lipid bilayer. So how come there is water inside the cell? Even though water is highly insoluble in the membrane lipids, it readily passes through channels in protein molecules that penetrate all the way through the membrane. Many of the body's cell membranes contain protein pores called aquaporins that selectively permit rapid passage of water through the membrane. The aquaporins are highly specialized, and there are at least 13 different types in various cells of mammals. You might think that the passage of water-soluble molecules and water itself can be difficult, but that is not the case. Take the example of red blood cells. The amount of water that can enter inside red blood cells is more than 100 times its volume. Other lipid-insoluble molecules can pass through the protein pore channels in the same way as water molecules if they are water-soluble and small enough. But if the water-soluble molecules get larger than the average water molecule, the rate of diffusion of them gets slower. Take the example of urea. Urea molecule is 20% larger than water and diffuses a bit slower than water due to its large size. But urea can still penetrate into the cells within minutes. So, there are two ways through which diffusion occurs in water-soluble molecules. Through pores and channels. Pores are composed of integral cell membrane proteins that form open tubes through the membrane and are always open. Pores are highly selective and only allow selective molecules to pass through. For example, aquaporins permit rapid passage of water through cell membranes, but exclude other molecules. Aquaporins have a narrow pore that permits water molecules to diffuse through the membrane in single file. The pore is too narrow to permit passage of any hydrated ions. Channels, on the other side, can be selectively permeable and contain gates. Many protein channels are highly selective for transport of one or more specific ions or molecules. This selectivity results from specific characteristics of the channel, such as its diameter, shape, and the nature of the electrical charges and chemical bonds along its inside surfaces. Now, the gating of protein channels provides a means of controlling ion permeability of the channels. To understand gating, let us get back to our example. Both sections of the bus were separated by a gate or this barrier. Now this gate can be opened or closed. This gate can be opened or closed in two ways. Either the bus driver has some button to open it that sends electrical signals to the gate. This will be called as voltage gate. And the other method will be that driver uses its hand to open the gate. If we consider the gate as channel, so the opening and closing of channel will be in two ways. Voltage gated and ligand gated. In voltage gate, the molecular conformation of the gate or its chemical bonds responds to the electrical potential across the cell membrane. Strong negative charge on the inside of the cell membrane may cause the outside sodium gates to remain tightly closed. Conversely, when the inside of the membrane loses its negative charge, these gates open suddenly and allow sodium to pass inward through the sodium pores. This process is the basic mechanism for eliciting action potentials in nerves that are responsible for nerve signals. So, what about the ligand gates? Some protein channel gates are opened by the binding of a chemical substance, a ligand, with the protein, which causes a conformational or chemical bonding change in the protein molecule that opens or closes the gate. One of the most important instances of chemical gating is the effect of the neurotransmitter acetylcholine on the acetylcholine receptor, which serves as a ligand-gated ion channel. Acetylcholine opens the gate of this channel, providing a negatively charged pore about 0.65 nanometer in diameter that allows uncharged molecules or positive ions smaller than this diameter to pass through. This gate is exceedingly important for the transmission of nerve signals from one nerve cell to another, causing muscle contraction. So, let us have a small summary now. We discussed about diffusion. Then, two types of diffusion. In non-facilitated diffusion, we have lipid-soluble diffusion, which was direct, and water-soluble diffusion that has further two types. Pores and channels. There were further two channels that were voltage-gated and ligand-gated. And we are still left with facilitated diffusion. Let us talk about this in the next section now.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.